Hello, everyone, and welcome to phyloseminar.org. This is the first talk in a series of 3 talks on SYNteny and its connections with phylogenetics. Please use the YouTube live comment box to ask questions. Today's speaker is Nadia El-Mabrouk. Nadia did her PhD with Maxime Crochemore at Université Marne-la-Vallée in France before joining the Mathematical Research Center at the University of Montreal for a postdoc with David Sankoff in 1998. She's presently full professor and graduate studies director at the Computer Science Department at the University of Montreal. She has long-standing experience in developing algorithms for comparative genomics and more specifically genome rearrangements, gene tree reconstruction, and tree reconciliation. Nadia is involved each year in the program committee of some of the most popular conferences in computational biology, such as ISMB, Recom, WABI, and others. She is regularly involved in the organization of bioinformatics workshops, the latest being Novel Mathematical Paradigm for Phylogenomics, held last summer at the Banff International Research Station. Welcome, Nadia, and thank you for participating.
Thank you very much. Thank you for the presentation. Thank you for the invitation. So my presentation will address different problematics related to the small parsimony problem. I will review some progress and work by my group on the inference of ancestral gene contents. So understanding how a gene family, a set of genomic regions, or a set of related genomes have evolved from a common ancestor is an important step towards elucidating many biological questions. If a rooted phylogenetic tree is already available for the considered biological units, represented at the leaves of the tree, then the problem reduces to the small parsimony problem, which consists in inferring the content of ancestral nodes in a way maximizing a certain parsimony or minimizing a certain cost or probabilistic criterion. So I will first recall the classical small parsimony problem, first on the gene sequence level, then on the genomic level. I will then focus on our recent work, first a new algorithm for inferring ancestral gene content, minimizing gain and loss episodes, then a more comprehensive evolutionary model for genomic regions, also involving duplications and horizontal gene transfer. Synesth, so this algorithm, is a generalization of a gene tree, species tree reconciliation, accounting for segmental duplication and losses. Then I will say a few words about FullSynesth that take as input a set of gene trees rather than a single SYNteny tree. So let's first recall the classical small parsimony problem. So given a tree topology, such as this mammalian tree with labeled leaves, the small parsimony problem consists in predicting the labels of the internal nodes based on some criteria. For example, leaves can be labeled with gene sequences, and the problem is then to predict ancestral gene sequences. This can be done by aligning the sequences, considering each column of the multiple alignment as one character, and use the classical Sankoff or Fitch algorithm to infer ancestral characters in a way minimizing, for example, here the number of character substitutions in the tree. So if we apply the Fitch algorithm, these are here the set of nucleotides that give an optimal score for the tree. If we choose A's at each internal node, this gives the optimal cost of 2 for the tree. As you see, we have one substitution here from A to G and one here from A to T. And of course, we have to do this for each column of the multiple alignment. I'm sorry. A little bit. Okay, so this was for the gene sequence level. Now on the gene and genomic level, in the genome rearrangement field, the leaves are rather labeled by ordered sets of genes, and the small parsimony problem consists then in inferring gene orders at internal node of a species tree. Predicting the evolution of gene content and order rely on models and involving content-modifying operations, such as losses, duplication. As you see here, the red, the green gene was duplicated, and on rearrangements events modifying the order of genes, such as inversions, transposition, translocations. So this is a full example, not a full example, but a part of an example taken from a paper we published in Molecular Biology and Evolution in 2015. It represents the evolution of tRNA gene content in bacterial genomes, in bacillus genomes, actually. So each genome is represented as an ordered and signed set of genes, possibly with duplicates. Here, for example, two red genes representing here isoacceptors. And the problem is to infer ancestral genomic architectures in terms of gene content and order, minimizing a given distance. So optimization can be done on various things, for example, on the number of order disruptions, number of breakpoints, or symmetrically the number of adjacencies, or it can be done on rearrangements events, like it's the case here. Okay, rearrangement events and content modifying operations, so rearrangements as inversion, transposition, translocations, and content modifying operations such as duplication, the horizontal gene transfer, etc. So here, for example, to go from this genome architecture to this one, we can do one inversion. So this segment here is inverted, and then one duplication, one tandem duplication. Actually, this segment is duplicated in tandem, and then an inverted transposition. This segment here is transposed somewhere else and inverted and transposed somewhere else in the genome, and here a loss. Okay, so this is a full scenario involving all such operations. Okay, so this is part of the scenario we have published in this paper here. Now, this is not an easy problem. To get to this scenario, we used a heuristic and simplified assumptions, such as the fact that operations are non-overlapping. Okay, so in that case, we can express the problem of aligning two genomes as an alignment problem, actually.
So to be clear, that's just two, they can't be overlapping on a single edge. It looks like you have a lot of overlapping.
Yes. Yes, but okay, so this is an extension. This is a heuristic. The idea is to can have non-overlapping on two genomes. I can't go in details. Anyway, this is not my main subject, right? This, okay, ideally, we would like to have such scenario, but I'm going to simplify it then, to remove the gene order. But I wanted just to begin with to remind what we did earlier. So we can have some heuristics. In that case, this is a nice case because the bacterial genomes were not too close to each other to have differences on them, but also not far enough so that we still can figure out the operations and not have too many possible scenarios, actually. Okay, so our heuristic worked on this data set. But in general, all these problems are NP hard, okay? So even for pairwise genome comparison, so when we have just two genomes and we want to find the minimum inversion distance to go from one to another. Actually, when the two genomes are just a permutation, so no duplicates, no losses, so just inversion, there is a polynomial time algorithm to compute the inversion distance. But in general, when we add different gene contents, duplicates, everything becomes NP hard for pairwise, for even for two or even three genomes. So I'm not going to go in the detail. And on a tree, almost all models lead to intractable problems, even when restricted to the median, so a non-rooted tree with a single internal node. But as I say, with some hypotheses, we can have heuristics, etc. Okay, so I'm still in the introduction to so another thing that we can do is to infer the copy number at internal nodes of a tree. So this is another small parsimony problem. So another small parsimony problem would be to infer gene content, but ignoring gene order. Alors here, the count package developed in 2010 and still being improved by my colleague from the University of Montreal, Miklos Xyros, addresses this problem on gene family at a time. And it's just a count algorithm, okay? So no scenario of events, just count, gene count. So for example, if we keep here the gene family in red, okay, because as I said, the count software just take one gene family at a time, okay? So here for the red gene family, for example, we can figure out that we have one copy in this genome, two in this one. So we have the numbers on the leaves, and the count algorithm predicts the size of this gene family on internal nodes on this phylogenetic tree. Okay, so count software implements different models, the Dolo parsimony, Wagner parsimony, and also a probabilistic birth and death model accounting for different rates of gene loss, gene duplication, and gene gain. Okay, so this is actually a model allowing to infer the size of ancestral gene families. So it's a well-known software, well-used software. But again, gene families in this case are considered one at a time. Okay, so the work is done one family at a time. Okay, so in a recent work, actually a work that we have just submitted to Recom Comparative Genomic Workshop, we rather considered episodes of duplication and losses that can involve many genes in a single event. Okay, so as I say, this is a work we just submitted to Recom Comparative Genomic, presenting a software called in-out parsimony for the small parsimony problem, accounting for episodes of gain and losses. So for example, here is the input is a tree where the leaves are labeled by genomic regions, homologous genomic regions, or syntenies, but I will forget the order of the genes, so just consider the set of genes for each leaf, for the leaves of the tree. And the problem is to infer the gene content of ancestral nodes in a way minimizing a cost of gains and losses. And here, gains and losses are gains and losses of many genes at the same time, but without considering the order, just subsets of genes. Okay, so this is, for example, one possible scenario with 6 gains and 4 losses. So the gains are in dots here. And the losses are represented by x's here, crosses. Okay, but this is not an optimal scenario if we consider the unit cost. So if we count the number of scenarios, because there is one with 8 events, 2 gains and 6 losses. The 2 gains, so one big gain here and another gain here, and 8 losses. So this the more parsimonious scenario is obtained by grouping gains actually up in the tree and also grouping losses and postponing them lower in the tree. Okay, so how to find such an optimal scenario? Of course, we can test all gene contents, all possible gene contents at each node of the tree. Okay, but this gives rise to an exponential time algorithm if we try all subsets of the set of gene families at each node. Okay, of course, this is untractable, but rather we show that the entire solution space is covered actually by testing 4 cases for the content of each internal node, which leads to a linear time dynamic programming algorithm. So the 4 states of a node are defined according to 3 sets, mean, out, and in. So the mean content corresponds to the minimum gene content of the node for any valid history. So in brackets here, I put the position of the least common ancestor of all the, for example, A. This node here is the least common ancestor of the leaves containing A. Okay, we have A here. So the least common ancestor of A is here. For C, C is present in all these subtree, but is not in the other. So the least common ancestor of all the leaves containing C is this one. Okay. So for a given node, X min, which correspond to the minimum content at this node for any possible history consistent with the tree and this and the leaves. Okay, X min is the set of genes with LCA at that node or above that node and contained in the subtree. For example, A, B, their LCA is up this node, and they are both contained in this subtree, in the subtree rooted at V, and C, its LCA is at this node. Okay, and it's also contained in this subtree. So X min is A, B, C. So for any scenario, any possible scenario, we must have A, B, C at this node. Okay, and then we have also out. So the out is the set of genes, is the gene family that are not present in its descendant leaves. Okay, so D, F, G are not present in this in the subtree rooted at V. And now X in is the set of genes which are present in the subtree, but with an LCA lower than the node. For example, E here is in. Okay, so the in genes are the genes that are gained, that can be gained lower in the tree. They can be gained lower in the tree, but we can put them in the content of V to group to make the scenario better. Okay, so with these 3 sets, we can then define the 4 possible states of a node. I don't know if I missed something, I think. Yeah, okay. No, oh, sorry. So this one, okay. So it's easy to see that each node can be then in one of the 4 states. So either it's mean or out or in out, okay, depending on its content. And the dynamic programming algorithm tests then each case for V, considering each case for VL and for VR. Notice that the 4 sets here, the 3 sets, represent a partition of F, which is the set of gene families. Okay, so we can a node can be in 4 states. It can contain only the minimum, or it can contains genes from X out, or it can contain gene from X in, or it can contains both genes in out and in. So this makes 4 cases. And then we try all 4 cases here. And the dynamic programming tests each case for V, considering each case for VL and VR. This scenario, for example, correspond to taking the minimum content for V. Okay, this one, for example. Okay, yes, having AB here, for example, this scenario here correspond to taking the minimum for V and the minimum for VL and VR. Actually, it corresponds to this equation here in the dynamic programming equations. While this scenario here, for example, we can have this content at V. Okay, so it's not the minimum. We have AB, but we have other genes which are in the again in the in set. Okay, so this scenario corresponds to the state in for V and in out for VL and VR, and this is represented by this equation here. So the algorithm stands on several recurrence equations, computing the cost of a tree rooted at V, considering its state and all the possibility for its subtrees. For example, this equation illustrates here the case of V in state min and VL on state out. Okay. Okay. Alors, in this case, this scenario will not be possible. It's not minimum, actually. This not because okay, this scenario here with the loss here, because if this node here has additional genes that have to be lost because it's not in the leaves, we should have a loss in this tree here. So instead of so in addition, okay, so if we have to lose B, rather than losing here, we can put it in the okay, this is not optimal because we can group the loss of B and C together later in the history. Okay, so this would not be optimal. So in that case, in the case when V is in the state min and VL in the state O, we should the minimum doesn't have a loss in this branch, and this is illustrated by this equation, for example, here, where you see that it doesn't contain any loss here, where we don't consider the cost of a loss. Okay, so this is the way we can understand these equations here. So the first pass of the algorithm, okay, so in the first pass, we go bottom up and we compute each one vector of dimension 4 for the content min, out, in, and in, out for each node of the tree. For example, here, 1 corresponds to the minimum cost of the subtree rooted at this node here. If we take the minimum content, if we take content min for this node, okay, and the cost is 2 if we take as a if the state of this node is out. Okay, so you see, instead of trying all possibilities, we just have to try 4 cases, 4 states for each internal node. So in a bottom-up traversal, we compute all these vectors, and then a backtracking procedure up to down, illustrated by red pointers here, gives an optimal edge labeling. And edge labeling means the positions of the gains and the losses. Okay. Now we still need to have 2 passes through the tree to additionally get the content of internal nodes. Actually, 1 possible content of internal nodes. The content, okay. So the genes families in black and red, so the min and in genes are added during a bottom-up traversal of the tree, and then the blue ones, those belonging to out, okay, are are added during a final top-to-down traversal. But anyway, it remains linear. So this is a linear algorithm. Okay, so
sorry, before you move on, so obviously, like, can you remind me again the fundamental choice that you made in order to keep this from being an NP hard problem? It's that you're looking at the gene content and not like gene order and things like that. Or is that?
Yes.
Okay.
Why it's not NP hard? This is a very simplified version. Yes, we just group yes and gain and losses. So we don't have any rearrangement events. Also, we don't have a duplication. You see, the genes are, we don't consider paralogous genes in a genome, for example. We have one element per gene family for each synteny. So yes, there are many things that make it. It's a very different problem from the comprehensive one that I showed before.
But it's elegant. Yes. Okay, thanks.
Yeah. But this is a nice small problem, actually, and it's surprising that it have not been studied before. So it's I think it's an interesting extension of just minimizing of the count algorithm, for example, of Miklos. So actually maybe it can be adapted. The probabilistic approach developed by Miklos Xyros for its count for one gene family at a time can probably be adapted to this new scenario with grouping gene gains and losses. So I think it's interesting to actually we began, as I will just say after that, we began with a very complicated scenario. And then actually it was complicated. So we simplified it because we couldn't, in our complicated scenario, we couldn't take account for segmental gains in our cost function. Anyway, so I will go back to another more complicated scenario just after that. Okay, so just to see that it can be applicable, we decided to apply our software to a data set of mitochondrial encoded protein families called mitocogs that we have considered in a previous paper when we were studying the endosymbiotic gene transfers events between the mitochondrial and nuclear eukaryotic genomes. So we took the same data of 11 plants. The scenario obtained considering a unit cost model involves two large gains. Actually, first I have to tell you how to read this tree. So the light gray, you have two trees here, okay? So in the one in light here represent the mitochondrial tree. So the content of the mitochondrial tree. And the dark gray here represent the nuclear tree. Okay. Now each internal node of the mitochondrial and nuclear tree contains 7 rectangles representing these complexes here. Okay, studied in this Ghanaian paper here. Each rectangle is divided into strips corresponding to the genes of the complex, and each strip is colored when the gene is present at the node. Okay, so for example here, anyway, so I can't go there. So what we can see here, so the scenario obtained when we consider a unit cost model for gains and losses, so just minimize gain and loss episodes. So we get two big gains here. Plus is for a gain. Okay, when you see plus, it's a gain, and when you see minus, you see losses. Okay. Okay, while those get okay, so with our algorithm with the unit cost, we have two gains, while those gains are rather split into smaller ones with a previous model in which gains are simply the lowest. So, when we simply infer gains at the LCA of the leaves containing this gene. Okay, so what we can see is that this scenario avoids splitting the genes of a given complex into different gain episodes, which may be considered more biologically relevant. For example, the SDH complex here is gained in 2 episodes with the unit cost model, while it is gained in 3 episodes with the gains at the LCA model. Actually, what we wanted to see with the two, we wanted to see if the gains in the mitochondria, in the nuclear, actually was compensated by a loss in the nucleus. We didn't really see such thing from it's not very convincing, actually. I can't say that it's very convincing, but it's just an application of our algorithm, just to illustrate how we can apply our algorithm. Okay, so of course, there are pros and cons in grouping losses and gains in single episodes. In one hand, this can appropriately reflect the unique transfer of full gene complexes, but on the other hand, it can also lead to artificially large genomic sizes on internal nodes. So this argues for an intermediate model going beyond the count single loss model, but penalizing each segmental loss according to its size, for example. So an intermediate model between the counts, which consider just single loss for with one gene, to these big losses, maybe we can have an intermediate level, okay, which will be probably more interesting from a biological perspective. Okay. So let's go to my third part now. So now going beyond gains and losses, in a previous set of paper, we presented a generalization of the gene tree species tree reconciliation that we called Synesth, taking as input a species tree and a synteny tree. So a tree leaf labeled by syntenies and and giving as output one or many possible history, minimizing a certain function. However, to simplify, as I told you, so to simplify the problem, gains were excluded for the optimization function. So this is the reason why we then considered independently the problem of just minimizing gains and losses. And actually now we are ready to update our Synesth algorithm in order to incorporate gains in our cost function. Okay. So now I'm going to introduce this more general reconciliation model underlying our Synesth softwares. But for that, I will need to recall the classical gene tree, species tree reconciliation model. So let's take again the example considered previously for illustrating the small parsimony problem on a genomic level. Okay, so consider this species tree leaf labeled with these considered genomes. If we keep just the red gene family, we can represent the species tree this way, with each genome represented by its set of gene copies. Now, suppose we also have obtained from another data set, possibly a multiple alignment of the corresponding gene sequences, a gene tree for the gene family. So we have the species tree, we have the content in this red gene for the species tree, but we have also a gene tree, okay? So then this embedding of the gene tree into the species tree gives rise to a scenario of events. Here, duplications. So, for example, the presence of these two genes here can be explained by a duplication in this branch, the same in this branch, and here too. And here, the absence of the gene here can be explained by a loss. So this embedding gives rise to a scenario of events explaining the difference between the two trees. More precisely, given a gene tree and a species tree, a reconciliation of the two trees is an embedding of the gene tree into the species tree, allowing to explain the topological difference between the two trees through a sequence of events shaping the gene family inside its host species. So, say differently, the classical gene tree species tree reconciliation problem consists in inferring the relationship and evolution of homologous genes by mapping the nodes of a gene tree onto the nodes of its associated species tree. Okay. For example, this possible embedding of this gene tree into the species tree, so this an embedding, allows to label the internal nodes of these gene trees with the mapped with the species it maps to. Okay, so for example, this speciation here have occurred in this ancestral genome A here. Okay, so the reconciliation is a mapping of the node of the gene tree by the corresponding species and event. Okay, so circle these stuff are events. So the events that we can consider here are speciations. So circle correspond to speciation, squares to duplications, diamonds to horizontal gene transfer. So we in Synesth we consider possible transfer from one species to another by lateral gene transfers, and we have losses here represented by half circles. So this may be the best or most parsimonious scenario if we limit ourselves to the species X, Y, Z. But something which is important to notice is that we can have missing species. And if we take into account such missing species, we can have better scenario, okay? Because the species included in a study, or equivalently the absence of species that are not chosen, unsampled, or extinct, has a serious impact on the output of a reconciliation algorithm. Okay, so here, for example, the transfer. So if we consider let's say an outgroup that was not in our study, okay, and if we consider a possible transfer from unsampled or missing species, it gives a better scenario than this one. Actually, we have one less loss here. Okay, usually actually adding unsampled species allow avoiding to have many speciations accompanied with many losses. So for this reason, it's important to account for the possibility of transfers from unsampled lineages. So that's what we do in Synesth. It seems simple, but actually it's very much complicates the equations and the dynamic programming. But it's important to consider this possibility. Okay. Anyway, I was just talking here about gene tree, species tree reconciliation, so single genes, okay? Now we have to extend this gene tree, species tree reconciliation to SYNteny reconciliation. So tree reconciliation has been widely studied to elucidate the evolution of gene families, with each gene family taken separately. Okay, so this is appropriate for gene family evolving separately, but not to syntenies, which are segments of colocalized genes evolving together through segmental events. So it's important to generalize it to segmental events. So rather than considering, for example, if we have these two regions in a genome, so we likely have actually it's likely explainable by a duplication of the three genes one at a time, not one at a time, just as one event. Okay, so we extended reconciliation to a SYNteny tree, okay, which is a tree. So now we need to have a SYNteny tree, so where each leaf is labeled with a SYNteny, so where we consider here as groups of genes. So again, in our Synesth software, we ignore the order of genes in a SYNteny, just consider its content. Okay, so it's considered as a set of genes. Okay. So the considered operations here for our generalized reconciliation act on sets of genes rather than on single genes. So we can have the gain of a set of a subset of the alphabet, the loss of a subset of a set, the duplication of a segment, and actually we also considered the split of a SYNteny, so a SYNteny that can be cut. Actually, this can be explained by a duplication followed by a loss, but we can also, if we want, consider it as a single event. And we also considered in Synesth horizontal gene transfer, so the transfer of genetic material from one genome to another. We considered two types of horizontal gene transfer. One keeping the genes in the donor, which better reflects the conjugation mechanism, and one removing the transfer gene from the donor. Okay, so here, when CD is transferred, it's removed in this side here, which best reflects the transduction mechanism. So viruses moving genes from one cell to another. Okay. Okay, so actually, we can have many possible, okay, you have to know that the reconciliation, we can have many possible reconciliation. Actually, we can have an infinite number of possible reconciliations. So among the infinite space of possible histories, because we can always gain and lose the same gene many times. It's an infinite number possibilities for histories explaining the embedding of one gene tree into a species tree. But among the infinite space of possible histories explaining the evolution of a SYNteny tree inside a species tree, both given as input, we are interested in selecting the most likely ones. So a possible way is to assign costs or probabilities to event types and to select history minimizing or maximizing the overall cost or probability. But the resulting history can vary significantly depending on the chosen costs, and it's always difficult to fix the cost of a duplication versus a gain or a loss, etc. So rather, we can just consider event vectors storing for each gene for each history, its content of events of each type. For example, this history here has one duplication, two transfer, and one loss. Okay, this one has one duplication, one transfer, and two losses. Okay, we can notice that actually I ignored the speciation because speciation doesn't really allow to make the difference between two histories. So we usually ignore them in the count vectors. Okay, so these we can just keep this type of vectors. And then we can look for histories such that no history with a better vector exists. So what is a history with a better vector? We say that a vector dominates another vector if for each dimension its value is equal or greater than the other vector. But in this example here, for example, there is no one which is better than the other, because on this dimension here, this vector is better, but on this dimension, it's this vector which is better than the other. But if we had, for example, another history with two duplicates, two transfer, and one loss, we would have eliminated it because this is better than the other. Okay. So history with non-dominated event vector are said to be Pareto-optimal. And we can then get rid of an input set of costs, and then the problem becomes just to output one or all Pareto-optimal histories. So we use algebraic dynamic programming to progressively subdivide the history space. Alors first, to a finite by exponential size of Pareto-optimal histories, then to a polynomial size set of Pareto-optimal event count vector, and eventually to the single cost of an optimal history. We adapt the semi-ring structure of the recurrence relation to compute desired results. Roughly speaking, for any node V here of a SYNteny tree with children VL and VR, and any feasible mapping, so sigma for V, gamma L for VL, and gamma R for VR in the species tree, and for any event at V and any SYNteny X. So we have to consider many things. Okay, any mapping, any consistent mapping. Now any event at V and any content X, Y, Z on these three nodes here. We define the set of history starting at V, followed by two paths. This is one path and this is another, and followed by the history rooted at VL and VR. Okay, and as previously, as a first step, we didn't consider minimizing gains. Okay, so actually XYZ, we don't, as I explained it before, we don't try all the contents in genes, but we just have to consider three states. Actually, as I showed you, when we will add the gains in our optimization function, this would be four states. In, you remember, mean, in, out, and in, out. So X, Y, Z. It's not it's not all the possible subset of the set of gene families, but we have to just try four states for each node. Anyway, so this leads to a representation of histories as composition of subhistories and path, which is used to formulate the induced form of Pareto-optimal histories. So I will not, of course, go into the details. Okay, so we also have to consider all possible paths between two nodes of a SYNteny. And actually, we show that we can restrict to three cases for the paths. One case with no transfer, one case with only one transfer, and one with exactly two. Of course, okay. So the number of all Pareto-optimal histories is exponential, but we can obtain the number of such history in polynomial time. I go very quickly. Yeah? For a given cost function, by replacing the two operator of the semi-ring structure, we can then rather compute the cost of a minimum cost history. the number of minimum cost histories, or we can output a given minimum cost history. Okay, we can output such history, which slightly increases the polynomial time complexity. Of course, giving the number of optimal history is faster than giving actually the corresponding history. Anyway, so we can output more or less many whatever we want, and rather with another replacement of the two operators of the algebra, we can compute the number of Pareto-optimal count vectors and eventually one history for each. I think this can be interesting. Okay, so just to say that we applied the Synesth to real biological systems. We applied Synesth to CRISPR-Cas class 1 genes. The literature reports a classification of CRISPR-Cas system based on a multi-dimensional analysis. So this is the classification taken from this paper from Makarova here. So if we consider such classification as representing a synteny tree, which actually is not necessarily the right thing to do, because this is not based on phylogeny, but I'll come to it later. The problem becomes that of finding then an evolutionary history which can explain the inconsistency between this tree and the bacterial tree from which each synteny is taken from. Okay, so this corresponds to the input of our problem. Okay. So we just output a different scenario, taking the classification of class 1 CRISPR-Cas system given in, okay. So taking this classification, we recovered an evolutionary history which is broadly consistent with the scenario reported in the literature. Okay, and actually, when we don't consider unsampled branches, we have here a non-realistic scenario with many duplications, speciation, and losses. You see a sequence of many losses here to go to the synteny in Escherichia coli. Rather, when we account, okay, but to obtain, let's say, more realistic scenario, when we allow for transfer from unsampled species. Okay, so okay, I think I'm a little bit out of time, right?
Yeah, I think we should probably move on.
Okay, probably go. Okay, so I will skip this part. Okay, so in this part here, I was discussing the fact that we can't really go begin with a synteny tree, because what we have actually it's not synteny tree. What we have are usually is a set of gene trees. So we also developed another algorithm taking as input a set of gene tree and simultaneously building the synteny tree and making the reconciliation. Okay, so the idea here is to find the better synteny tree, the synteny tree minimizing the reconciliation cost. Okay, so I will move to the conclusion. Okay, so conclusion. So we can still have do many things, of course. On the part on episodes of gains and losses, we can extend the model to more general weights, accounting for the size of a gain or a loss. We can generalize our algorithm to enable exploring the whole solution space through algebraic dynamic programming. Right now, we output the solution, minimizing the size of the syntenies. We can also extend the model from the Dollo to Wagner parsimony, so allow for many gain points for a given gene family. On a comprehensive model for Synesth, we can integrate Okay, so this is the next step. It will be to incorporate what we now know on minimizing gains episode. We have to incorporate it in our algorithm. Of course, we can think about integrating gene order. So we have many ideas about that, but of course, the general problem is NP hard, but we can go further in some way in this field. Okay. Okay, we can also find solution to work on the solution space and output co-optimal solutions. And I will skip this because anyway, I didn't talk about my third part, etc. Okay, and this is my final slide. So the main students who worked on this last uh, uh, progress of my in my lab are Matteo Delabre and Mathieu Gascon. Matteo Delabre is my PhD student working on this algebraic dynamic programming. And I have to thank many other students that I mentioned the work in my presentation. So that's all.
Thank you very much. I'm impressed. Like, you've been working on these for a long time. The problems are so hard.
Yeah.
They're so hard.
Yeah, and I took so many time actually
But
to
it seems like
explain.
real progress is being made. I have
Yeah.
a technical question and maybe a more biological question, and then we should probably wrap. But what is algebraic dynamic programming?
Yeah, it's not a biology, but yeah, I try to explain on my best. But the idea is to express the problems in a way where we can combine different things, you know. For example, now with algebraic, we have these dynamic programming equations for the problem of answering the syntenies, minimizing gains and losses. And on the other side, we have these equation the better the species and events, and we want to combine the two. Okay, when you express them in an algebraic programming way, it makes it easy to combine them. Okay, so conceptually, it makes it easy to adapt. So it's a way. It's a given a way of expressing the dynamic programming equations. In a way, we can adapt it more easily. For example, to go from for example, outputting all Pareto-optimal, if we want vectors, to outputting that just the cost of the best solution. Okay, when everything is expressed in an algebraic way, you can easily just change the operators of the algebra to go from outputting all history to outputting all costs, etc. So it makes it more flexible. It's very hard. Well, that's that's
So, I mean, but the
the
essence is
idea.
you're just expressing your algorithm in like purely algebraic terms.
Yes, and yes, in that way you can just change. So, for example, so uh, going from outputting the cost to outputting the history, just you change just the operators of the algebra. Okay, so you go from a union to a plus or something like that. So it makes it easy to go from one problem to another, one output to another. So this is the very important work of my student Matteo Delabre, who is actually finishing his PhD. So if you know people interested in having a very interesting, very good postdoc, so he will be valuable in a few months.
Cool. And I mean, this is maybe a bit more of a more biological question. I mean, like, obviously the parsimony problem is very hard, but biology doesn't necessarily minimize this function. So, I mean, if you look at simulations that where
Sure.
that are using sort of realistic parameters, how often are they sort of parsimony optimal?
Yes, I can tell you. Yes, and yes, and it depends also what you are minimizing. If you're minimizing the number of, well, but it likes a probability too, because when you have better, you try to have the most probabilistic scenario. You need to know the probability of a transfer compared to a duplication, etc. That's why that's actually the reason why it's interesting to have such an algebraic expression, because rather than just outputting the most parsimonious, we can rather want to have all the how they like you need to have a representative by equivalent classes. Okay, so for example, one representative of the history with one transfer, one representative of the histories with je sais pas, with just one unsampled species, so we can better play with your space, your solution space.
Cool. Okay. Well, thank you so much. That was a great start to our session.
Thank you, Eric. Thank you to giving me this opportunity.
All right. So we'll see people next time. Thank you.
Okay. So thank you.